or absorb the chemical, or you're exposed to a bacteria. This chemical looked just like your own PDCE2. The chemical might have only been in your body a short time. It's almost like a hit and run disease. And over time, the immune system starts to mistake that reaction. It mutates, and it thinks the chemical is similar to yourself. And ultimately, it reacts with PDC2, and this may take years to develop. We like to think that this autoreactive cell that's giving all the trouble can be controlled and can be sent to hibernate. We can think we can do it in the mouse. Moving into humans is a lot more complex. With age, well, you start to react even more with PDC, particularly the PDC on what's called the blebs of your small bile ducts. And that's where the trouble begins. But ultimately, these cells start to hurt the bile ducts, and they produce inflammation. And the inflammation produces more inflammation. In other words, it can become a vicious cascade with symptoms like fatigue, visits, doctor tests, and oftentimes doctors get confused. I hear all the time stories of patients who say, you know, I went to the doctor for 5 years and nobody paid attention to me until I was finally diagnosed with PBC. It's not as common as it used to be, but it still occurs even now. There is, however, a bright side to this. We have now found the molecular sequence to many AMA antibodies. We got a really nice grant from the NIH to look at the real molecular definition of these. A number of these antibodies react to these chemicals or to bacteria. We think by deciphering the binding structure, we're going to develop some new therapies. And by knowing the structure, we may be able to figure out who's more susceptible. We're doing a lot of genetic fingerprinting of people. One of the things I've learned over the years, and what I'm about to say is not my quotation. I'm not sure who originally said it. I think it was a 19th century philosopher. But what they said was that the answers to every question are known. The problem is the person who knows the answer has never heard the question. And the reason I bring that up is our lab has been instrumental in developing animal models, in developing what we call recombinant antigens and clones and everything else. And we have given them away without charge. We even pay the shipping to anybody in the world that wants to study it. The reason is the more people that study PBC, the higher the likelihood someone's going to come up with the breakthrough that we need to cure this disease. And so, at least in my lifetime as a researcher, I've trained probably almost 200 scientists by now that have their own laboratories in Japan, in the UK, in France, South Africa, really just about everywhere. And a lot more people interested now than were interested when I started in the mid-1980s. So that's the first talk. You want to hear the second? Yeah, okay. So I got to get off the podium and move my computer. I don't want to fall off that. What causes PBC? We think everything I've told you before is that there are multiple events that have to be acquired. You have to have some genes that predispose you. You have to have some insult. I had a guy in my lab, and he did this to me. I didn't know this was going to happen, but what I say to people is that you get PBC because you have bad genes and bad luck. Like you get lupus, bad genes and bad luck. You get PBC because you have bad genes and bad luck. You can't do anything about your bad genes, but you can hopefully do something about exposures. What I want to emphasize is the immune mistakes I talked about, the bugs I've talked about the chemicals, whether it's pollution, chemicals, or toxins. But I haven't talked about hormones. And as I look around the room, it's pretty clear there's more women here than men. And it's even more important to note that there's more women with PBC than there are men with PBC. And by the way, there's more women with lupus than there are men with lupus. So why is that? So we spent, I don't know how many years and how much money developing mouse models of PBC. Mice that looked like they have PBC. But in those models, it affects the male mice and the female mice equally. And that's wrong. We would like a model in which the females in the mice are the ones who get disease, because it can explain the female predominance. It might also give us a really important lesson about PBC. Clearly, females have an intrinsic abnormality that makes them more predisposed to autoimmunity. In fact, women have at least a 5-fold increased risk of developing autoimmunity than men. What's the explanation? It's not just estrogens, by the way. It's true that female hormones are involved, but it's not the only explanation. What produces the gender bias? And how is that going to lead us to new therapy? What causes the skewing? Well, we have 23 pairs of chromosomes. Men have an X and a Y, and females have an X and an X. Is it possible that it's on the X, and women have twice the X? And there is some basis to suggest that's true. And in fact, hormones and the number of children you have all can affect that. But it's not the only answer. Maybe women are exposed to certain things that men aren't exposed to. And that's certainly true. But that's not the only answer either. And if we're really going to ever solve PBC, we need a model that's a faithful model. And don't misunderstand, we've been delighted by the animal models we've made. And the NIH has given us hundreds of thousands of dollars every year to help us develop those models. But always they sort of whisper, but they're not female predominant. Because if you want the same physiology that you have, we have to know the explanation for the female predominance. And we have to be able to look at the men with PBC and say, well, what's special about you? Even though you're male and have PBC. The models have to be predominantly female. They have to produce AMAs. They have to have lymphocytic infiltrates. They have to affect the bile ducts. And they have to produce cholestasis. All of us are basically born with the same genes. But as we get older, they undergo something called somatic mutations. However, when activated by something, how the levels are produced are different between men and women. And that should be a clue. What does that mean? Why are people different? Why, when you immunize women with influenza vaccine did they respond differently than men with influenza vaccine. There's a pathway which is known as the interferon pathway. All of us have it. Interferon is a really important molecule for inflammation, for autoimmunity, for cancer, and so forth. And a number of these genes become hyperactivated in women compared to men. But they really haven't been studied very well in PBC. And it's possible that some of these genes become super aggressive and they change. And this molecule called interferon is what I call very pleiotropic. It can affect cells all over the body. I'm building you up for the grand finale in just a few minutes to explain where I'm going and why it's important. And there are multiple genes and multiple families that can occur. And so here's where I'm leading to. I told you earlier the answers to all questions are known. It's only that the person who knows the answer hasn't heard the question. Well, life is full of coincidences, what I call stochastic events. And I happened to meet two scientists from NIH. One is named Dr. Howard Young. And Dr. Young told me about a mouse that he was studying in his lab. And he studies interferon. And the thing about that mouse is that it was producing too much interferon. He had manipulated this mouse for other reasons. It's what we call a designer mouse. All of us produce interferon. What Howard did is he made a mouse in whom the interferon stays and survives longer after it's made. So like you take aspirin or you take any drug, it gets metabolized and it's gone within hours. He made a mouse in whom the interferon, when it's made, lasts for days, maybe even for weeks. And he told me about it, and he also told me something that just made my imagination go wild. He said, you know, he says, the mice scratch. And he says, they change the pigmentation of their skin. Well, he thought the mice had lupus. And in fact, they may have a mild lupus, but it's not particularly important or dramatic. But as the mouse got older, it was the females who got the disease, not the males. And in fact, the males seemed to be protected. And more important than that, his mice produced AMAs that looked exactly like the AMAs you have. Under the microscope, their livers look just like the livers of women with PBC. And it's only the females. It's not the males. The designer mouse has a mutation of the gamma interferon gene that allows it to have chronic expression. You can see the females have a higher level. Look at the far right, the levels on those two bars. One is almost twice as high as the other. And if you look under the microscope, they have histology just like you would see in PBC. And they get cholestasis, just like people with PBC. And their bile acids go up, their bilirubin goes up, they get inflammation, they scratch, and they make high-titer anti-mitochondrial antibodies that have that same word you couldn't pronounce, autoepitopes, as people do. And this has all happened in the last 6 months. Once again, a very serendipity event. I happened to meet Howard Young. He told me about his mice. Obviously, a little more complicated than that. Takes a while. Things are done. He sent us the mice. We have them now in Davis, California. He still has his mice at NIH. We did extensive studies on the mouse. We wrote it up as a scientific paper. It's just now coming out. It will be in a few months in hepatology. They asked the hepatologist from England to write an editorial about this work. We hope that this mouse is the beginning to developing new therapies. We can answer why females are developing it. And we're now in the process of raising money and applying to NIH to study these mice. Because here's what we think. We think that people with PBC have an overexpression of the same gene, gamma interferon, or something we call downstream of it. And you may be, what's that? You mean downstream? A downstream, D-O-W-N? Oh, gamma. It's the Greek word, G-A-M-M-E, G-A-M-M-A, gamma. It's a Greek symbol. Interferon, I-N-T-E-R-F-E-R-O-N, interferon. I was once a spelling champion in the 3rd grade, but I think that was in 1952, so I may have misspelled it just now. But we think that people with PBC, perhaps what happens goes something like this. You get exposed to a chemical, you have a genetic predisposition, or you get an E. coli urinary infection, you make a reaction to it. But the reaction isn't very high, and it might even disappear over time. But then you might have an incidental infection, a viral infection, bacterial infection, who knows what, a cold even. And that raises your level of interferon, and it boosts the response. And in people with PBC, you may produce even more of it than you should. And that keeps going up and up. So those who have this chronic but asymptomatic autoantibody in their blood, ultimately it tips over and you get disease. And that disease produces inflammation, and then the disease perpetuates on its own. And it becomes important for a number of reasons. It means that we may need designer therapies. We may need different drugs for different people at different stages of PBC. The drugs we may use in early diagnosis may not be the same drugs in people that have more advanced stage. And that's a really important point. It also might give us a clue as to how to treat the inflammation that this interferon is producing. So we have high hopes that this model is going to attract industry who want to study their own new drugs to hopefully come up with a cure for patients with PBC. It's long overdue, I can assure you of that. Anyhow, I hope this wasn't too much over the top. And as always, it's a pleasure to be here. I'll be around all day. Anyone that's come to the conferences, you can walk out in the hallway, ask me anything you want. If I can answer it, I will. Thank you very much. And obviously, I'll answer anything you'd like right now. Sure.
Okay, while we're waiting for those questions to come forward, I'll do some housekeeping kind of stuff here. You've all been a great audience today. Just a little reminder that the feed is not going directly into the video camera, so any room noise will be recorded, so we really have to focus to keep it to a minimum. Oh, it fed now? Good. And if you should have to leave the room, please duck. We don't want your head on the camera. You will have nice hair, but we don't want to see it on our videos. So we have people also that will be starting to do my favorite job and hit you up for money. We have 50-50 raffle going, and then the tickets for the Chinese raffle. A lot of the items in the Chinese raffle were Intercept employees did a thing and raised money, and they went out and got the gift cards that are back there. So they're kind of contributing in more ways. And there's some great prizes back there. Tickets are $1 a piece, 13 for 10. We'll be drawing the winners of those items tomorrow at lunchtime. The 50-50 raffles, one each day, and you'll be picked, the winner will be picked in at the break in the afternoon. So, okay?
Oops. All right. I'll load my slides and get back. Oh, you want it now for the next speaker? Oh.
And as long as I'm up here, I'll take care of this so I don't have to climb the stairs again. It's a very small for what he does. As you can see, in addition to all he does in the work, he's been running the projector for us today and helping us with all our technical difficulties. So he's Superman up here. And this is just a very small token of our appreciation.
Oh, aren't you
Dr.
nice?
Gershwin.
Thank you so much.
Thank you. Want me to hold those and hand them to you, or you just want to pick
I'm
through?
fine. I'm fine. I'm sure this will be lost by tomorrow. I'm the guy that loses iPads, iPhones. You have no idea. That's why I have well, we'll go into some other time. Okay, let's take some questions. Can you be AMA positive without developing PBC, or eventually? And the answer is yes. First of all, you should know the AMA in most hospitals is done by what's called immunofluorescence, and it's not a terribly specific test. So you have false positives. So we urge anybody that's not sure of their diagnosis to ask their doctor to get the AMA performed by a lab called INOVA, I-N-O-V-A. INOVA has the recombinant assay. It's the only test approved by the FDA for the diagnosis of PBC with recombinant technology. Okay, INOVA, I-N-O-V-A. If it is a real AMA that's positive, you are at higher risk, but it's not 100%. With the increasing use of, I need help, wrong glasses. What is that? Increasing use of marijuana. Sorry. My glasses, sorry. Recreation and other things, how does this affect the PBC liver? I don't think there's any data that suggests that it hurts the liver of PBC, and I doubt that it helps the liver either. You could ask that to Dr. Vueling, but as far as I know, there's no significant effect. Okay, naltrexone research. Well, there are a number of people that are trying to use that, naltrexone and others. I'm not convinced that there's really any benefit. Ask that to Dr. Bolus tomorrow. I think he knows a little more about naltrexone than I do. Another one. Well, eating foods that are anti-inflammatory help PBC health. In other words, eating right, is that going to help? Well, you know, in theory it should. In reality, I think it's like putting out a fire with a water gun. I think it's really the other way that might hurt you. Eating diets that are too rich in fatty foods, that are too rich in oxidants. I think